Hello, everyone, and welcome to Better Biopharma. I'm your host, Tyler Menichiello, and I'm very excited to be joined today by Dr. Nicole Paulk, CEO and founder of Siren Biotechnology. For those of you who don't know, Nicole is a pioneer in the gene therapy space and currently serves on at least a dozen different biotech scientific advisory boards. Her company is developing an off-the-shelf AAV gene therapy platform to treat multiple solid tumor types. Nicole, thank you so much for being here.
Happy to be here.
So, for the folks at home who aren't familiar, would you mind briefly describing Siren's platform?
Absolutely. So this was spun out of my lab at UCSF where we wanted to kind of ask a pretty audacious question, which was like, A, could you use AV outside of kind of Mendelian monogenic genetic disorders in oncology? And B, could you make a single AV that could be used to treat more than one thing? And so this started from a project that we lovingly called the Universal Payloads Project. And fast forward like a decade worth of work, we have Siren. It was really to kind of do those two things. It was to see if we could make an EV that could produce or that could package a payload that we could use to treat more than one thing. We went with engineered cytokines and decided to point the ship at oncology and kind of looking to see whether or not you could use AV in an oncology space. Since, right, fun fact, not everyone necessarily knows this, right? AAV is not an oncolytic virus, right? So this is not like adenovirus or measles or polio or some of the other viruses that folks have thought of when they think of viruses in the oncology realm, right? This is a non-oncolytic virus. So it itself doesn't do anything with anti-cancer, but it doesn't mean that you couldn't use it in a cancer context. And so really wanted to kind of address those kind of two big worlds firsts. And spun out of UCSF back in, oh goodness, I think I started it on paper back in 2020, and I think we spun out in 2021, and I resigned in 2022. And we came out of stealth in 2023. So basically, once a year, we have a big milestone. Uh, is apparently how that goes. And we're off to the races and still preclinical, but going to be clinical stage next year.
That's very exciting. Thank you so much, Nicole. Um, so I really wanted to talk today a lot about when we had our pre-call. You mentioned how Siren is switching from plasmids to producer cell lines, and I was very curious what prompted or motivated that switch, and if you could kind of describe what made you realize that plasmids weren't your way forward.
So I think for the vast majority of folks, plasmids are still going to be the way for at least the short term, right? Because they are so cost effective and because they're so fast to kind of queue up and ramp up. Um, the challenge I think becomes that like over time although they are cheaper to kind of initially set up, they are more expensive over time. And so the unique kind of aspect of Siren, the fact that we get to make one single AAV, but it could be used to potentially, treat, dozens or hundreds of different things, is that we only need to go through like process optimization, that whole process one time, not, dozens of times for each of our different assets, for each of our different programs, right? We've got one asset to date, right? Siren 101, but has the ability to go after many things. And so for us, we still have the same cost sensitivities that everybody else did when you were like a seed stage company where you're like, gosh, we have like single digit millions in the bank and need to do something very quickly. So it still makes sense to use plasmids initially. But for us, like once we show clinically that Siren 101 has legs and it's got a nice safety profile and looks like it's going to have some efficacy as well, then it absolutely makes sense for us to switch over to producer cell lines. Because again, we're only going to be making Siren 101, no matter if we're going after high grade glioma or lung cancer or breast cancer or kidney cancer, it doesn't really matter, right? We're going to be using Siren 101. And so for us, it makes sense to make that switch over to producer cell lines and really kind of never look back and really only be using plasmids for other new innovative things that we're testing out in the lab for maybe our second universal asset and these types of things. And so I think for many folks, the decision has been a difficult one to make about when to make that transition because producer cell lines are still more expensive upfront and have a kind of a bigger time hurdle upfront. But I think as that continues to be innovated on and come down, I think other folks will start to join Siren in moving and making that switch over to producer cell lines for their kind of bigger assets that are going after bigger indications, even if it's still only one program at a time. right? Because many other folks are starting to pivot into bigger diseases like, diabetes and these types of things. So I think you're going to start to see this shift where there's definitely going to be companies still working. Like you're probably still going to be in plasmids if you're going after a very, very ultra orphan, ultra rare disease. But for folks who are starting to make the pivot to go after much more common indications, I think you're going to start to see folks also join in the PCL kind of decision framework tree about when do we switch and what's that decision process like, and switching over to PCLs.
Yeah. Thank you. And this might be a naive question for you and those of you out there who aren't familiar. I studied biology, but I was never a staff scientist. I've never worked in pharma. So forgive me if this is naive, but you said plasmids in the long run as you scale up are more expensive?
Yeah, right, because you are going to need right, you're gonna need those plasmids for every single batch, for every single run. At GMP, those become very expensive.
Mm.
Um, Bless our hearts, we love E. coli and they do great things for us as they produce us plasmids, but they are very expensive producing plasmids at scale, right? When you start needing enough plasmid to produce, 5,000-liter bioreactor runs of AAV, that starts to become very expensive.
Yeah.
Media that they need, the antibiotics that they need, the purification that they need, right? It's a whole separate, it's like a drug within a drug, right? That also needs to be GMP if you're using that for your GMP lot. And just you take any reagent, it could be a tube, it could be a plasmid, it could be a cell, it could be media, it could be coffee, right? Whatever, delicious coffee or whatever we're talking about. If it has to be GMP, like just 10x the price minimum, maybe 100x, maybe 1,000x. So anything that you can remove that has to be GMP, um, from kind of like your list of things that you must buy, um, uh, just massively improves on cost, right? You need a minimum 2 plasmids. Most folks are doing triple plasmids, so it's going to be 3 plasmids at GMP, each independently run with all their own specs and all their own release assays and their own master cell banks and all of their own, all the documentation and paperwork. And it's just like if you can just replace that with a single, GMP PCL line, that's a lot less reagents and paperwork and master cell banks and separate supportive reagents and kind of consumables and things that you need. It just makes things faster, easier, cheaper. It's like the thing that you always want but you never get, right? You always want, right, the impossible triangle: faster, cheaper, and easier, but you only can pick two. um PCLs are actually the ability to get all three once they're established. So once you have them, developing them is still expensive and does take time. So you're going to have that upfront hurdle. But if it's something you are going to make a lot of, or you're going to need to make repeat batches of, right, like a universal AAV gene therapy asset, then it absolutely makes sense and it will be like way, 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 way cheaper in the long run.
Yeah. No, that makes sense. It was funny. You said you love E. coli, and I thought, you could say that in a room full of biotechnologists, and nobody will bat an eye, but try saying that on the subway, you're gonna get some funny looks probably.
So sad. Like at an infectious disease conference, you'll get.
Yeah, right. Professing your love for E. coli. So in retooling your process to be producer cell line dependent, I'm curious if there were any unexpected challenges in shifting over that you might whether that you expected or didn't expect.
So we are in the midst of doing this as we speak. So we haven't completed the shift yet. So we are still using plasmids and are developing our PCL basically as we speak right now. So far haven't encountered anything. It is moving along as planned. And the point for us where we would switch would be after our phase 1 clinical trial. So after our phase 1 clinical trial shows that we've got some nice safety, and that Siren 101 is going to have like a path forward clinically, like to me, that is the moment where we make the switch from plasmids to PCL, because we would need to make a larger batch at that point anyway for like the phase 2/3 trial and for commercial. And so that would be the moment that we would switch and then probably never look back for Siren 101. So we'd make the switch to PCLs and drop plasmids for Siren 101, but probably would still initiate plasmids for like a second asset. So if we queued up a second universal asset that had some unique cookie that made it able to go after something that Siren 101 couldn't, we would still probably queue that up with plasmids as well, because you just can't beat the fact that like you could start tomorrow, and then it's just deciding when do you make that switch. Is it while you're preclinical, or would you do it after your clinical stage? And so that still a corporate decision, all of the things, but So still room for both. I think there needs to be both for all eternity, plasmids and PCLs. It's just when for your company does it make sense to make the switch, if ever. And for Siren, I think for us it's after our phase one.
Great, thank you. So when we were talking earlier, you told me about this CDMO Hunger Games, if you will. Why don't you tell the people at home what the heck I'm talking about?
So we did something pretty heretical here in Silicon Valley. For folks who don't know, our seed round was led by Founders Fund, right? Like the most premier of the premier of the premier, like tier one tech investors, not biotech, tech investors, right? In like I think Founders Fund's history, I think they've made like 10 biotech investments ever, right? They don't typically invest in biotech, but when they do it in if it's a company they have a lot of conviction in. And so like normally when you like get money from a tech VC, right? They want to know like how many years until you are a decacorn. It's like no longer cool to be a unicorn anymore. You have to be a decacorn, babe. It's like how long until you're going to be a decacorn? And I did this heretical thing with our seed funds. I went and told my partner Scott Nolan at Founders Fund. I was like, Scott, I'm going to use almost all of the seed money that you gave me to run essentially a CDMO Hunger Games. And he's just like, what are you talking about? And I was like, so here's the plan, right? I sit on, like you said, I don't even know anymore, probably at least a dozen, probably more,
Too many boards.
advisory boards, many companies. And if I'm not already on your scientific advisory board, I often get brought in as an external KOL when you go on CMC hold or clinical hold, help you try to get off these things. Almost all clinical holds are, in essence, also CMC holds. It's almost always for a CMC reason. And so it almost everything ties back to manufacturing, whether you did it in-house or whether you did it externally at a CDMO, it doesn't really matter. They're all CMC. And so I have seen every possible reason why you can go on CMC hold or clinical hold, and they're all manufacturing related. And so I have seen over and over and over again how many different ways you can muck up manufacturing and how it can really, it can really be harmful, not only for your company, like financially, but for the timelines, for the patients, for safety, for all kinds of reasons. And I did not want that to happen to Siren. And I was just like, you know what? Rather than kind of like pointing and guessing and wondering, like, well, was it this that caused it? Or was it this that caused it? Or was it this that caused it? I was like, let's be scientists about this. Let's like break it down and let's just find out what matters and who matters and like run a very big experiment. Let's take like a GLP-esque lot of plasmid, like our GOI plasmid, our gene of interest plasmid, and let's send it to everybody. Everyone gets like an aliquot of the exact same run of plasmid, right? So let's send some to every single CDMO basically in the US, and basically tell them we are going to run a competition. We told everybody about everybody else. I'm not going to name everybody here, but like we told everyone about everybody else. Right? We sent it to you. We also sent it to your 10 biggest competitors. Right? Everyone knows the names of all of these folks, so I don't need to repeat them here. But like we essentially sent an identical volume and lot and concentration of plasmid to everybody and said like the clock starts now. We are going to have you guys fight and produce AAV for us. And we want you to do it the same way so that it's fair. We want you to do like a 10-liter suspension production of AAV and HEC293 cells using like kind of a very standard run-of-the-mill process so that we can compare like what is like how long does it take you to turn this around? What's the yield? What's the potency? What was our interactions with the team like? And, kind of what were the deliverables? And did we like the reports that you gave us? And did we like interacting with your team? And what was the legal experience like negotiating this agreement? And then actually take the vector that we get back and actually run experiments with it and test these things head to head to head and see how they perform and just kind of really get down to brass tacks and compare not only like all kinds of scientific metrics that we were interested in, but also kind of, corporate metrics that we were interested in terms of cost and turnaround time and legal clauses in the agreements and all these types of things. And so we had this massive matrix of like all by all things that we wanted to compare. And we sent it to everybody. We told everybody that we were doing this and that it was a competition and just kind of said like, all right, like the ball's in your court. The clock starts now. Let's see what you can produce. And then started to get all of that vector shipped back to us. And it was unbelievably illuminating It was probably the single most important experiment that we will ever run at Siren that I can unfortunately never publish, and can never really talk about, with actual data, because all of the data from all of these is, confidential, these types of things. You have to sign, unfortunately, you have to sign a confidentiality agreement when you do one of these things. But I can talk about it in broad strokes. And it was probably the most important experiment we will ever run at the company because it was not close. Right? Like it was not like, oh, everybody was really, really close to each other and we could have picked any of them and they all would have been fine, right? There were very big differences in all kinds of parameters, from maybe things that only matter at a corporate level, like price, but also maybe things that matter at a scientific level, like the yield or the potency or the level of contaminants and impurities and just all kinds of fascinating and interesting things. And so some of those might not matter to you if say you're doing really, really low dose local infusions in like maybe an inner ear gene therapy program. Doesn't matter to you what the yields are because you're going to use so little. But if you're doing an in vivo intravenous infusion for like a skeletal muscle disorder where you're going to need a lot, right? That might matter a lot to you, right? And so all of it depends on who you are and what your program is and what you're going after and what your needs are for that particular program and that particular disease and those patients. But like the spread was enormous and on all of these various lenses and all of these various metrics. And so for us at Siren, like it was very obvious, like for us and for what we were working on in our indication space and these types of things, what we needed. And this is the one name I can name because it's who we went with and we put out a press release together. So we chose to go with Catalent. And it was just very obvious that they were going to be the absolute best CDMO for us. But that experiment, which is like, can you imagine like telling a Silicon Valley tech VC that you're going to like do a bunch of CMC optimization experiments with the money that they gave you for the seed round? Like imagine a more, and I say this with all the love in my heart for my CMC homies, like imagine a more boring experiment that you're going to pitch to a tech VC. Like we're going to do a bunch of CMC optimization, which me as an AAV person, I get very excited about, but it's somewhat outer sun. Can you imagine something more boring? But that is exactly what we did. We spent almost our entire $6 million seed running that CDMO Hunger Games. And it was probably the single most important experiment we will ever run. We did it when we were like a 3-person company. We existed like barely even on paper yet. And I'll never get to publish it and I'll never get to talk about it. But probably the single most important experiment we'll ever run.
I absolutely love it. That's one of the funniest things I've heard, and it also makes perfect sense. I could only imagine the reaction on the other side when they got this letter and it's detailed. We're in the Hunger Games. What? So
Here. But like what's crazy too is that like it allowed me to like see into the future. Like I could see into the future 6 years. Had I gone with this particular process or this particular CDMO or this particular manufacturing style or this particular whatever, like, all the combination of things that go into someone's platform for how they produce, yada, yada. Had I gone with, vendor X or something that had a very different profile than what we went with, I could see 6 years in the future. And I'm like, if we had gone with this, like this would have absolutely potentially like led to a CMC hold like 6 years in the future when we were like, now running our phase 1/2 trial, back when we ran this experiment, starting in 2021. It's 2025 now, so we're already 5 years in. I could absolutely see that being a thing. So it's like I impress upon folks, not everyone is going to have the scratch to run it. That was a very expensive experiment. We spent almost all of our $6 million seed doing that. It was very expensive. So I realized many folks are not going to be able to have the scratch to do that. But I urge folks to maybe do a little bit of that, and to just not, right, don't just take the advice of like your board. Like maybe you got your seed investment from, whoever capital, random Joe Schmo capital. And they're like, oh, well, one of our other portfolio companies uses X. So you should too. They're great. And they might be great. They might actually be great for like that company, for their program, for their specific thing, for their capsid, for their, route of administration, for their specific disease space, but it may not be great for you. Right. And so like, I encourage folks, don't just take at face value like this is a famous name, or this is they've made vector for this other famous group and they're doing well, or they're based in this country and so are we, and so it'll definitely be good, like whatever. I encourage folks to like be a scientist about it and really, you know, have the data. And you can never trust on someone else's data. So don't just trust what's in their brochure. Like, this is the normal yields that we get with this capsid. Like, no, 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 no. Like, you got to run this experiment yourself. Maybe you can't afford to have 10-liter productions. Maybe it's 2-liter productions. Maybe it's shake flask productions. Maybe it's whatever. Um, but I encourage folks to actually go through the process, and it will take an inordinate amount of time and money, but in as much as you can. Maybe you can't do it with 15 of them, maybe you can only do it with 3, but like have data drive that decision, not just like the advice of a board member, or who has the shiniest brochure, or these types of things. And it will pay dividends in the long run, because you won't end up going on CMC hold and clinical hold, hopefully, right? You might still, for some other reason outside of your control, but like you can at least have as much data going into it as possible, driving that decision, so that you can feel confident that like you're making a good choice. And maybe the choice you make is like, I'm going to make it myself in-house, or like whatever, but like you want to have data be driving those decisions, not just like the advice of a board member, or some like purely corporately strategic metric, like cost. Because it's almost always who AV is a you get what you pay for product. Cheap AV is usually cheap for a reason.
Yeah. I mean, finances aside, it's kind of the most common sense approach to CDMO shopping I've ever heard, to be fair. So how many did you have participate in this competition? You said 10, 15?
I think it was up near 10. It was up.
Wow.
Don't quote me on the exact number, but I think it was up near 10.
Yeah. It's interesting that you say there was a pretty notable difference across the board. Like, I was gonna ask you, like, what would you have done if they all came back equivalent value? And you're like, "Oh, well, I don't even know."
I mean, it's like if everything scientifically was the same, like the yield was the same, the potency was the same, the turnaround time was the same, the levels of contaminants and impurities and anti-full ratios and all of these types of things, and that how well they actually performed when we used them in experiments. If all of that had been the same, then it would have just resorted back to like, more fuzzy wuzzy things, like
Yeah.
who did we enjoy working with more, right? Right. There were big differences in terms of like the folks on the teams that you got to talk with and meet with. Like, who did we enjoy working with more? Whose reports were more clear? What was the cost? What were the legal terms like? Right. There's very big differences in terms of the clauses in some of these agreements. And kind of those more kind of corporate and fuzzy wuzzy considerations would have come in, had it been a perfectly level playing field, in terms of what we got. But there was no like every single CDMO had differences on every single one of those lenses. It's not like every one of them was bad across all lenses, and everyone that was good across all lenses, right? It was a completely, heterogeneous smorgasbord. And so that's what I mean, where I say like it will be different for every company. Like, if you are an inner ear gene therapy company, you have very different vector needs than someone who is doing intravenous administration for a skeletal muscle gene therapy. Going after an adult indication is going to be very different than going after a pediatric indication. Something with local delivery versus intravenous administration, something with an original capsid versus an engineered capsid. Like all of these things play in. So it's not that there's one great CDMO and everyone else is bad. It's that it will matter and be different for you and your company and your programs. And so um, you really kind of got to run the experiments yourself, to see what is going to be best for you and your programs, and kind of what you're doing.
Yeah. I think we would both agree that you can never underestimate the importance of the fuzzy wuzzies of
For sure.
all of it. Now, this is the last question.
When stuff hits the fan, and it'll eventually hit the fan, for some reason on some lens at some point, maybe not now, maybe it's 4 years from now, maybe it's 8 years from now, you're running your phase 3 and you're going commercial or whatever. But like, how well you get along with that team and you communicate with that team, and what's your transparency like, and how much do you trust them, and these types of things, right? We always talk about corporate culture culture, and like, how much do you like your culture, right? How much do you like your vendor's culture? Are they going to be able to retain their top talent? Because like you want the team that's making your phase one GMP lot to also be the team who's making your phase like 2, 3 GMP lot, who's also making your commercial GMP lot. And so you want those good people that you love to be happy and to be retained, right? You don't want a lot of turnover. So it's not just what's the company internally at your company, what's the culture like at the really key vendors for you, right? The groups that are providing your animals for your animal studies, and the groups making your plasmid or your vectors, right? It matters. Um, so to me, like, you don't just go and audit those companies for your GMP run to see like, are they, crossing all the I's and dotting the T's, and everyone's signing off on the bottom of the page that, they witnessed that, yes, 50 milliliters of media was poured into the thing, signing off. But like, how do they get along? Do you get the sense that that team that you like is still going to be there for your next run?
Yeah, that's great. I do want to, as much as I would love to keep asking you questions about the competition, I want to move on to, in the sake of time here, to a little less fun topic, a little more depressing, but it's no secret that funding is been better, to put it lightly.
Better.
I'm curious to hear your thoughts on what the near future of cell gene therapies looks like during this time of declining investor sentiment.
I think, right, there's a little bit of, it probably depends on where you're at in your funding cycle. Like if you're cash out as a company in the next 3 months, you're going to have a very different, like, you need to do something very different than a company that's cash out 2 years from now. Um Versus, same thing, like if you're super early stage in your discovery stage versus your IND enabling preclinical stage versus your early stage clinical versus your late stage, all of these types of things. So it all depends on where you're at. Um but I think broadly, I think in the near term, I hope folks don't need to fundraise, because if you need to fundraise like in the next couple of quarters, I think it is going to be challenging. We kind of need things to like calm down and settle down, and everyone to go on summer vacation and like go get a tan, relax, and then come back in the fall. Um, Not that big financings happen in the fall, right? Fun fact, financings happen from JP Morgan to 4th of July, and after 4th of July, for the most part, things get pretty sleepy for the second half of the year. VCs go on vacation for the summer. They come back in the fall to negotiate very small agreements. Uh, Usually like bridges and mezzanines and other types of things, and to help their existing portfolio companies negotiate like partnerships with big pharmas that they can announce at JP Morgan in January, and then they get sleepy again after Halloween because now it's Thanksgiving and Christmas, and everyone goes on holiday before JP Morgan. So it's like JP Morgan to 4th of July is the window. So hopefully you don't need to raise before JP Morgan, because you probably won't. And that's not just cell and gene. That's really, I would say, just say biotech. So like, hopefully you don't need to raise before January, because you probably won't. But if you need to raise in January, like, you've got to differentiate yourself from plain Jane vanilla. Like, what are you doing that is unique and that is novel and that is exciting? And my prediction, I might be wrong, but my prediction, I'm willing to stake my stake in the ground and say this, right? There's always a cycle between platforms and assets, right? It's platforms are hot and then they're not hot. Now it's assets, and then it switches back and forth, and they always exist on some cycle. Sometimes it's 2 years, 3 years, 4 years, 5 years, but like, it's cyclical. Platforms and assets, back and forth, back and forth. We just left, I would say, about maybe 2 years ago, a platform cycle, and now we're in assets. It's single digit assets that have near term, like, meaningful milestones and readouts, that can drive some type of liquidity opportunity, whether it's an M&A from big pharma, whether it's an in-license or an out-license with upfronts, whether it's an IPO, yada, yada, these types of things. Um, and that is absolutely in vogue right now, because investors are so nervous because they haven't had a lot of blockbusters. They haven't had a lot of liquidity opportunities. They haven't had a lot of successful um, commercial launches, these types of things. All valid. But I think when we switch into the next platform swing, which I think is about a year out, would be my guess. I think we're going to continue to be in this very asset-centric um, pocket of time for, I would guess, about another year. But when it switches back to platforms again, which it always will, platforms and assets, I think this next one will be fundamentally different from anyone we've ever had. Because I think it's gonna last longer. I think it might actually last forever. I think this might be the last asset, and this is my super controversial hot take. This is the tea you came for. This is Nikki's. Maybe she's wrong, but if she's right, oh boy, this is my hot take. This is the last asset cycle we're gonna have, and that's because of AI. I think in this world, uh, where the expectations are that you need to be iterating faster and doing more faster, and differentiating yourself and having more things de-risked more quickly with a smaller team, with like a much leaner kind of capital usage. I think you can only achieve all of that with AI and with a smaller team and being capital efficient. But I think you can only really do that with platforms. And in terms of like the iteration times that are needed and these types of things. Doesn't mean you don't have individual assets. You still have assets as a company, but you must have a kind of a platform behind it. And so I think moving forward after this asset cycle, it's going to be a platform cycle, and it might be a platform cycle that stays forever. Because I think if you are like a single asset company going after a single indication, and I need $30 million to go run my Series A, and then after I do my Series A for my clinical trial, I'm going to queue up my next asset, and it's going to be from scratch. And we have to completely start from square one at discovery stage, and it's just like those days are gone. Like the days of needing $2 billion in 15 years for each one of your assets and each one of your programs, and they're all one for one. Like those days they're just gone. Um, so like everyone needs to reframe their models and like how like this is not just like entrepreneurs and companies. This is VCs as well. We need to kind of start from scratch when it comes to how we do biotech investing and what are the expectations and how much money am I going to give you and what are you going to give me for that amount of money and what are the deliverables and what are the timelines. Um, I think this next switch back to platforms is going to stick.
You heard it here first, folks. Platforms, platforms, platforms.
Platforms, assets for at least the next year, and then it's going to switch to platforms. Is not coming back.
Get the assets while they're hot. Wow, that's
But, like, plan for that. Like, if you believe me, if you think I'm not completely insane, which is it's a valid thing. But, like, if you think there's even 1% of me that's right, like, plan for that. Plan a year from now, platforms are gonna be in vogue again. How are you gonna be ready for that? Assuming like, how can you stay alive first? Like, how can you fund yourself to be alive through that moment? And then what are you gonna do to be ready for that moment, when it comes?
yeah. I was gonna ask you what your advice would be on how companies can innovate in this climate. Clearly, you're saying it people should be investing in platforms. What and forgive me if this is a silly question, but, to those companies out there that are focused on assets and don't really have a platform, they're in that traditional model of one for one, one on, one off. What is your advice? Like, how do you go about changing your gear, your philosophy, your strategy on like how can companies retool their philosophy, I guess, and approach to these?
I mean, you can do it internally, right? There's nothing that says you can't pivot, right? Like that is the definition of like a good entrepreneur is like you pivot with the market. Um, it's harder in biotech, right? We're not like, an app where you can just like change at the whim of your customer base. But it doesn't mean it's impossible, right? You can pivot internally. It also means like you don't have to do everything internally. Like you can partner with someone who has a platform and now like you can implement it into your workflow, right? You don't have to do everything in-house. It's not just like, oh, I have to, you know, outsource my manufacturing. Right. You can outsource some of your innovation, right? Not everything has to be done in-house. You can in-license things. You can do partnerships. You can find, one or two or three or four other really small companies just like you that are all maybe like about to go under. But like together you have enough money that if you all merged, right? We always forget about that M in M&A. And we always kind of assume that only big companies can do that. Well, only a big company can merge with like this little dangling itty bitty company that's so small and meaningless, but we want this one asset of theirs. So we will merge with them rather than acquire them. Everyone forgets about the M in M&A. They only think about A. It's only acquisitions, right? What if you merged with like one or two or three other really small companies and like maybe you each only have $5 million in the bank and it's not enough to do a clinical trial. But if you all four of you merge, now you got 20, like you can get a clinical program through for 20 million. And what if one of you has like a cool, innovative, some kind of platformy-esque thing that now you can put at the front of your pitch deck, right? Now you've got enough money to run a clinical program, you've got a platform, you've got a number of assets. And like now you're ready for JP Morgan in January. So like don't forget about the M in M&A. It is also available to you, and you don't have to wait for someone to come to you. Even you as a small company can offer to merge with another small company. These things don't have to be insane. You don't have to let someone else always be in the driver's seat. And also just don't be afraid like to get scrappy. Like you can do stuff in-house that is not, you know, it doesn't cost $10 million to innovate. Like you can come up with something cool, I promise you, for like hundreds of thousands of dollars. You're not going to get like in vivo NHP proof of concept data, but like you can get something that like shows promise that you can put in a pitch deck, that you can get folks excited around. It's possible. It's you just gotta not right. It's scary when you are facing going on like you have less than a year's runway and now some wackadoodle chick is saying, I want you to queue up one part-time hire to to like innovate something. So that you'll have something to show at JP Morgan in January. Like maybe that's a terrible idea for you, but maybe it's not. So like I encourage folks to just like take off right your preconceived notions hat and just like go into a room with a whiteboard and just like map some things out. Like just take all of the preconceived notions about what you have to do um out of the picture and just kind of chill out, have a beer, and like in front of the whiteboard and just like think like what are things that we could do that sound interesting and exciting and that would differentiate us and could potentially bring in new investors, right? New does not just mean new, you've never had them before, but it might mean it's not biotech, right? What could you do to bring in a tech bio investor or a tech investor or a family office or a sovereign wealth fund? Or, right, also, you do not have to only get money from VCs. There's grant money out there and all kinds of other sources of money. Like, get creative. Um, Call every one of your vendors today and just be like, I would like 80% off.
Won't happen if you don't ask, right?
Hey, with a very straight face. Most of them are gonna balk at you, but like maybe you come down to 50% off or 40 or 30 or whatever. But like do that with every single one of your vendors. Every single one. Every single one. Just do it today. Um, Even if you have a signed agreement, this big, huge, long legal agreement that you signed that's 500 pages. Okay. Still, just ping every one of your vendors and be like, I know that we agreed to this, but like I would like to pay this instead. Uh,
I have it or no?
Like, let's talk. You'd be surprised how many don't want to lose you as a client. They also need funds. They're also scared that you're going to go under, and don't want to lose you. And how many are willing to not only give you a discount or completely renegotiate an already fully negotiated, signed on the dotted line agreement, because they also don't want you to go under, and they also carry they also care a lot about patients and want to see that whatever cool thing that is that you have going, want to see that make it to patients and see the light of day. So like just negotiate everything.
It's terrific advice.
Everything. Just don't take anything at face value. Negotiate everything.
I'm gonna try that with my electric company in hopes of get down my god awful electric
First
bills.
thing to do is go tell you to go punch rocks.
Right. Or leave me without power, but but thank you though. That was great advice. And I for one am pro merger. Just imagine how many fun names. Don't forget about the imagine how many fun names we could have if all these companies start hybridizing and
Yeah.
making new biotechs.
If there was ever a time that you could do like a wackadoodle thing that like previously people would have been like, oh, I can't believe they did that. How weird or how how funky or how whatever, and like put their nose up at you. That was like in the past. That was like during the ZERP days. Like in this era where like everything is crazy, um,
Nothing makes
like,
sense.
the attention spans are 5 minutes. Can you remember what you had for lunch 2 days ago? Do you remember what was on the news cycle a week ago? Like, I can't no one can remember anything. Like, the attention spans are 5 minutes. So just, like, do something crazy. Now is the time. No one will remember a year from now. If it works, like, you're off to the races. If it doesn't work, no one will remember. So just do the cool thing.
Amen. Preach. Um, Along that vein of, again, low funding, tough environment.
Yeah.
We were talking before about a shifting organizational structure across the industry, and you said something to the effect of it's the death of the build versus buy debate.
Yeah.
Yeah. So, what would your advice be to young biotechs trying to navigate this shift and the shifting environment with outsourcing? And what would you say to anyone with convictions about internal manufacturing?
I think in this exact moment in cell and gene therapy, the idea of pitching to your board, um, if you are private, and to your shareholders in your board, if you are public, that you want to build an internal manufacturing facility is insanity. Um, like I cannot fathom a situation where uh where that would make financial sense, timeline sense, scientific sense, corporate sense. And I've heard all of the explanations like, but we do this very, very complicated thing, or we have a very, very unique something, something, or we want to own the full tech stack, or we want to have control over our timelines, and it's this laundry list of things. And I'm just like, yes, and like that's great, but you really wanna ask, investors for, at the low end, $150 million, at the high end, a half a bill to build something somewhere? Who are you gonna staff it with?
Right.
There's, like, no people. Um,
Tough sell.
I'm not gonna name names, but there are some fairly prominent, companies who have recently tried to buy up all of the available, like, CROs and CDMOs and CMOs and all of these groups, to try to consolidate them, and, like, that's not going so well. Um, so, just there is no space, there is no staff, there is no there's no money, there's no capital to do this. And I don't actually think it is going to save you that much time, and that much money and that much gray hairs or anything. I think that decision needs to that needs to die. That needs to perish. That needs to never be brought up again. I am absolutely wholeheartedly, and I'm not saying this as a shareholder or a biased party. Um, I own no stock in Catalent's or any other CDMO or CRO or CMO that we work with. I am just an advent believer that like in this era where I think you need to be a million times more capital efficient than we've ever been before with leaner teams and all these types of things. Um, I think the days of everything must be in-house are just dead. And not just for your manufacturing, but really on kind of almost any front. You should only have in-house the things that only you can do that are uniquely and supremely and only you. And I think everything else needs to be outsourced. Do you give up something to do that? Of course you do. You give up some amount of control and some amount of your timeline and these types of things. But in this environment, unless things go back to like ZERP type days, which I don't see them doing anytime soon. I just don't see those things coming back. I think you are going to start to I don't think we're gonna have the same thing that we talk I'm here in Silicon Valley in San Francisco, right? The fun party conversations are like, who's gonna be the first billion-dollar company with a single employee? Like, who's gonna be the first to do it? Someone's gonna do it. Who's gonna be the first? Right? I don't think we're gonna get there in biotech where you're gonna have a billion-dollar company with a single employee who outsources everything. But I think that that like exercise of thinking about it isn't far off from how you should be thinking. It's like, how can I do like 10 times more with 10 times less? And I mean that in terms of space. I mean that in terms of people. I mean that in terms of money. Uh, how can you do more with less faster, while still maintaining exceptional safety and efficacy for patients, so that we can move things much more quickly. And I think we are starting to see some shifts at the FDA that hint that they are going to start to try to implement some more creative and aggressive things to allow folks, to not only do things internally at your company, right, at the sponsor level much more quickly, but also on the FDA side in terms of review timelines and these types of things. So I think we are going to enter a world single digit years from now where like the 2 billion 15-year classic package for an asset, will go away, and that you could bring something from 0 to 1, right? The classic P or TL 0 to 1. You could go from 0 to 1 on a drug, I think, in low single digit years, and for hundreds of millions of dollars, not 2 billion. So if you believe that to be true, and you think you've got it in the legs to survive that long, right? What can you do to kind of prepare as a company? And so I think folks need to start like having these exercises in your head about like, do we need a head count of 200 or could we do it with 20? Do we need 5,000 square feet or do we need just, like 2,000 square feet? Do we need in-house manufacturing or could we outsource everything? Do we need to raise, does our Series B have to be $250 million or could it be 30? Um, do we need to raise VC money for the first 3 years of the company or could we do it all with grants, and delay that need to bring in VC money, um, and kind of take that dilution hit and all of these types of things. So I think like we are all going to need to reframe our thinking around like what does it mean to be a therapeutics development biotech company, that's making drugs, because I think like the carpet is about to be pulled out from underneath us and things are like changing underneath us as we speak, and I think that's a part of why it feels so uncomfortable right now, and everyone, you can kind of feel it. It's just kind of everyone's a little itchy. It feels uncomfortable. You're like things are different and things are changing and none of us like change and we're all kind of uncomfortable right now. I think part of the reason you feel that uncomfort and that like sadness when you see all these layoffs and like all of these things, a lot of that is not just like companies making big mean decisions. It's that like the world is shifting underneath us as we speak, and like you can do more with less. And so like think about how you can implement that and do that and like be in charge of it, right, rather than letting it just happen to you. How can you take charge, and kind of think about that from first principles and implement it in a way that's going to be most efficient for you and your company.
Yeah. I mean, you make a bulletproof argument, and I think that is all great advice. That kind of led into the next question I was going to ask you about leaning out operations, but you pretty much hit the nail on the head there. Uh, Along the vein of money, though, everybody's favorite subject,
Yeah.
cost of goods. It's no question, cell gene therapies are notoriously and infamously expensive. Um, I'm curious what innovations or paradigm shifts you see on the horizon that may get us to a point of significantly reduced COGS,
Yeah.
and what aspect of development and manufacturing should innovators be focusing on for the sake of efficiency and reduced cost of goods?
Yeah. And I think this will be different for every company, right? It's the classic AAV answer. I'll answer as AAV, but it's true for lenti, it's true for adeno, it's true for cells, right? I'll answer for AAV, right? Like what, and I always use these two examples because they are so different. Like if you are an inner ear hair cell gene therapy company going after like deafness or something versus like a skeletal muscle gene therapy company, going after some skeletal muscle like atrophy disorder, right? You could not be more different as an AAV gene therapy company. Like the inner ear hair cell company needs single digit microliters of AAV that they are able to infuse, like one, like 1 microliter of AAV, into these inner ear hair cells, and they're going to deliver it locally, right? Versus a skeletal muscle gene therapy company that's got to do intravenous administration in order to get perfusion throughout the entire body to hit all those skeletal muscle myofibrils, right? Could not possibly be more different. We got intravenous versus local, high dose and high volume, high concentration, or versus low dose and local, and these types of things. Right? Could not imagine two more different companies. They need to focus on very different things. If you're that intravenous skeletal muscle gene therapy company, almost nothing matters more to you than dose administered, right? We need to bring the dose down because you have to give so much intravenously in order to hit all of those skeletal muscle myofibrils throughout the entire body. Almost nothing else matters. You need to bring your total dose down. So, what are the cost drivers that are going to allow you to bring total dose down? It is probably not like, I'm going to add a new enhancer to the media formulation that's going to allow me to get like 0.2x improvements in yield, but aren't going to allow me to bring my dose down. Like, you as a company probably need to spend really any amount of money, uh, like as much as you can, whether it's internally creating a novel capsid that is liver detargeted and that is skeletal muscle tropic. So whether you are doing this internally with a capsid development team in-house, or whether you are in licensing, whether it's non-exclusive or exclusive, a capsid from any of, there's gobs of them now, numbers of fantastic capsid engineering companies. So whether you're doing it in-house or you're purchasing something from someone, you probably need to spend all of your time, money, focus, and effort on getting a new capsid that does allow you to administer a lower dose. That is probably the single most important thing you need to focus on, versus you're an inner ear hair cell gene therapy company focusing on treating something with local administrations with low doses in the inner ear. For you, you are limited by volume. You cannot administer more than like 1 or 2 microliters into the space that you're going after. So for you, you need things that are more concentrated, right? So you need things like, now it matters for you, like potency per microliter. For you, it matters that like you can get stuff that can be concentrated to very, very, very high concentrations without crashing out and aggregating. So like focusing on media optimizations and diluent optimizations such that you can pack and pack more and more AAV capsids into a smaller volume without them aggregating, without them crashing out of solution. You need absolutely no contaminants and impurities because you don't have a room for there to be even any. Right, because it's just like you have 1 microliter to work with. You need every last microliter of that to just be like AAV. There can be like as minimal diluent as possible and anything else that's in there, right? So you need to focus on a very different set of like biochemistry problems than a skeletal muscle gene therapy company. And so like knowing where you fall and what you need to optimize and what are the big drivers that you should really be focusing on, I think are important. And I can't tell you how many companies I either advise or I indirectly advise or I hear of or I see give presentations at big meetings or, what have you, where they are just laser focused on the wrong thing. It's like this is a thing you can optimize, but this is not the big driver for you. And so I think folks need to just really kind of like holistically think about like, what is the most important thing for my type of company and the types of things that we go after and the things that are the biggest drivers for us, that we can both innovate on internally, but also pay to have innovated externally by professionals who know how to do this kind of innovation and optimization. And don't be afraid to pay money for that, right? Like it might matter a lot, right? Like, yes, you might have to spend $100,000 to run this pilot optimization experiment, but if it keeps you from going on clinical hold, which will take you a year to get off and cost like clinical, like maybe you're burning 5 million a month, like that's like a normal burn rate for like a clinical stage company. That $100,000 is going to seem cheap in comparison. So like do the optimizations and all of that kind of work upfront. I promise it will be cheaper in the long run, which is obviously easier to say now and harder to do later when maybe you only have $2 million in the bank because you're still a preclinical stage company. But right, in as much as possible, like look into the future, look into your crystal ball, and like see what are the problems you are going to have. Look at what problems other companies and other sponsors and other groups are having, and like map that onto yourself. and think through like what are the things that I need to optimize and really spend time and money on now, to improve our cogs. And hopefully, all of our brothers and sisters out there who are not therapeutics companies, but who are working on novel reagents and novel instruments and novel manufacturing processes and the CDMOs and like all of these groups, we can start layering all of their innovations on top of the things that we're doing right internally. And together, I think in single digit years, we genuinely can. I know we keep saying this as a field and it feels like it's forever away, but like it really isn't that far off. We are single digit years away from being able to produce these things at scale for a far, 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 far cry from the amount of money that has historically been the kinds of cogs that one can expect that have led to list prices such that the 5 most expensive drugs on earth are cell and gene therapies. Um, I think in single digit years that will no longer need to be the case, but that is both a blessing and a curse. It is a blessing as a company because you won't need to raise as much money. It is a curse because you still need to convince your investors who are used to plugging into their little NPV model, click, click, clack, click, click, clack. No matter what, they're going to have a list price of $1 million. Well, what if your list price isn't $1 million? What if your list price is 150,000? Does the model still work? Do they still want to invest in you? Right. So we all need to kind of reframe. Right. Everyone's expectation. I promise you, if you find any investor on earth, not just US investors, any investor on earth that invests in cell and gene therapy right now, built into their NPV model, one of the assumptions at the top of their little formula is that this will have a list price of north of $1 million. But what if that isn't true? Right. So we all need to reframe expectations that, like, in single digit years, we will be able to have cell and gene therapies that do not need to have a list price that high because the cogs are so much lower. So how can we still make it so that there's enough of a margin that everyone gets a healthy margin, healthy returns, taking care of shareholders, but also making sure that our list prices are not things that get us hauled in front of Congress, and can continue to be paid for on things like Medicare and Medicaid and these types of programs.
Yeah, that was terrific. Very well said. Thank you. Um, we're at the pretty much at the end here, so I want to ask you the hallmark question of the show that I ask every guest. And you knew this was coming, but Nicole, in your opinion, looking at the industry or in AAVs and your niche within that world, how can we better biopharma?
I think particularly in this moment, I think one thing I would love to see everyone do, no matter who you are that's listening, whether you are a bench scientist, whether you're an executive, whether you are a journalist, whether you are someone who works at the FDA, whether you are someone who works, at CMS, everyone, like, just don't be a Debbie Downer right now. Like, don't be negative. Like, we all feed off of each other, and if all of us do is complain and say like the market's bad, and there's been failed launches, and things are expensive, and someone had a delay in their review timeline at the end, like, if all
Wah, wah, wah.
of us ever do this kind of piss and moan and complain, then we all just stew in it, right?
Yeah.
You are who you surround yourself with, right? Everyone enjoys being with someone who's positive and fun and happy and charismatic and all those types of things. And that extends into everything: how we talk to each other on podcasts, how we talk to each other at work, how we talk to each other at conferences, how we put out press releases, just everything. Just like, make the choice that, like, I'm not going to be a negative Nancy and only kind of piss and moan and complain. Like, choose to find positive things. There are absolutely people getting cured of diseases right now, and like, share those stories, share things that are hopeful, share things that are exciting, share things that are innovating, and just like, it starts with one person and it cascades. Because if you complain to me, then I'll complain to the next person, and then, like, all of a sudden we're all just complaining all the time. um there is no reason we can't lift ourselves out of this. We don't have to wait for, like, interest rates to drop and for something to happen at the macro level with the stock market and with Jerome Powell and all. Like, no, we can lift ourselves out of this. Like, we all have that power, and I empower and implore everyone to just, like, share positive things that are happening in the field right now, because it's a lot and it's a really exciting time.
I could not agree more on the whole attitude first front. That's a phenomenal point. A great note to end on. I would advise everybody out there in the Take Nicole's advice. First, get a whiteboard. Second, grab a beer. Third, grab your partner in the company. Figure out your platform, reduce your cogs, and after a couple beers, start sending out those DMs to other CEOs. Be like, hey, you want to merge?
And then ping all your vendors for 80% off.
Yes. And do it all with a smile on your face.
Smile on your face.
Thank you so much, Nicole.
No worries.
Thank you so much, everybody out there, for tuning in to Better Biopharma, a product of Bioprocess Online. We'll see you next time.